Okay, so today's March 29th, and I think that sets us at about 5 weeks to the exam, if I'm not mistaken. AP Bio happens to be one of the first exams this year in the AP season. And I'm gonna try to cover units one and two today, but that also means that in the subsequent meetings that we have, I should be able to get through pretty much all of the units. And I think if if at school you guys are taking AP Bio and you're doing units seven and eight, that might be pretty fresh in your mind. So instead of doing a review on that, we might have an opportunity to look at some questions together. And I'm not sure if anyone's had an opportunity to solve any of the 2013 AP Bio MCQ samples. I know that it's dated, but the questions themselves that still pertain to many of the topics that is still within the curriculum are actually really good. So that's something that we can do, and of course look at FRQ as well. Yeah. So, yeah, and if you have access to AP classroom questions, that's really fantastic. I think part of what I want to do today is to not only go over some of the content that you need to know in a relatively quick manner, but also because this is a review, it's a really great opportunity for us to connect some of the ideas that we're seeing here in units one and two to some of the broader concepts that we see in later units. Because most of you guys are probably at unit seven or eight, in your school curriculum. So that means that this will not only be an opportunity to review the first two units, but also see how the linkage exists between units one and two, and of course the rest of the units, particularly unit three and unit four, and so on. Okay. All right. So yeah, it does seem like most of you guys are pretty comfortable with chemistry. So I'm going to just kind of breeze through this, but I do want to make some couple of points that I think are really important and themes that I think will help you in solving some of the FRQ later. And you guys all know that everything that we find in biology are still part of the matter that make up the universe, and there's carbon, hydrogens, and oxygens, and so on. However, there's this one drawing that I always like to emphasize when I teach this in school, which is to say that when you look at sodium, this is part of the alkali metal. I think it has an atomic number of 11 for those of you guys who are in AP Chem. And you guys would remember that this is um, it's a very reactive metal. So when you put this into water, it will spark. And, as you go down that family, it gets even more and more violent. But it's a very explosive metal that you put into water. And then here we have chlorine, which typically exists as a diatom. That is to say that we have chlorine gas, and this is generally going to be very, very noxious. It's a toxic gas. But when you combine them together as sodium chloride, I mean, this is just salt that we put on our foods. And the main point that AP biology is trying to make with slides like this is to talk about a quality that we call emergent quality or emergent property. I'm going to type that into the chat as well. And in fact, that's what I'll try to do. Just some of the main terms that you might want to remember. And if you guys remember what emergent qualities are, is these properties that arise as a result of the combination of the substrates that don't exist when they're separate, right? Because sodium chloride, when you put salt into your soup, it's not going to explode into noxious gas. That's clearly not going to happen. And the main idea is that as the sodium atom and the chloride atom or chlorine atom come together in that ionic bond, it alters the overall structure of that substrate in such a way that it no longer acts like either sodium nor chlorine in their individual parts. And the reason that this is so important is that this is sort of the first time that we are introduced to the idea of how structure, I'm sorry, I'll rewrite that, structure and function are inseparable. And we see this all the time. As I was just talking about before with this AP biology FRQ, and I'm going to switch over to that right now. Again, I hope that this is going to be covered in my recording because I don't see it in my Zoom preview. But if you guys can all see this right now, the very first part of the 2023 AP Bio FRQ says, describe the effect that the addition of a charged phosphate group can have on a protein that would cause the protein to be inactive. And they're not necessarily asking about the specifics of any particular protein or any very, very detailed chemistry relationship here. All they're saying is, as I mentioned, when you add a phosphate group, that alters the overall structure of that protein because it's going to impart chemical interactions to that protein that's going to change the way that it behaves chemically. And that change could potentially turn a protein from an active state to an inactive state. And of course, we see this vice versa as well, especially when you look at signal transduction. Phosphorylation cascade is the idea that you attach phosphates to activate a bunch of proteins. So we definitely see this main idea being mentioned sort of earlier. It's almost like a preview of what's to come. It's like when you watch a movie and there's like a shotgun in the living room and you know that about 45 minutes into the movie it's going to be used. That's sort of what they're trying to do. And that's kind of what we want to always think about when we're studying for AP Bio, especially reviewing these units. Let's see. Anything else to be said here? So hopefully you guys are all going to be aware of the elemental compositions of different macromolecules that we see later. So that would be carbon, hydrogen, and oxygen for all of the macromolecules with differences in nitrogen and sulfur and phosphate. But um, one question that I can tell you guys, and this is sort of my opportunity to tell you guys some of the MCQ that I've had the opportunity to see over the years, is that you can see a question like this, and they may ask, there are lots of different types of elements in the universe, but it appears that all of the living things share the exact same composition or relatively similar composition of oxygen, carbon, hydrogens, and nitrogens. Now, they may ask why this may be the case. And there are two different ways that AP could sort of extract answers from you guys. One particular direction is more along the lines of like cosmology. And that is to say that when you look at the universe, the most common substances are things like hydrogen, helium, oxygen, nitrogen, carbon. Maybe not in that particular order, but you can ask ChatGPT. They'll tell you exactly what order of abundance they may be in. And when you take a look at what's happened in abiogenesis, that is to say, how living things arose from non-living things, at least in the scientific purview of things, we know that it's going to be opportunistic, utilizing what is most available in the environment. Like imagine an already unlikely event, like the formation of life, having to use some really obscure, very, very low in abundance metal in order to make it. It doesn't make any sense, right? So it's always about what is most likely, and these are going to be what's most abundant and available on our planet. Now, when we look at a more biologically relevant explanation as to why all of these elements appear in a very similar composition, would have to do with the universality of life, which sort of links to what we've been talking about. That is to say that we believe that the last universal common ancestor, the original precursor to all the living things that we see currently, well, we probably used these particular elements to make all the macromolecules, and we just essentially descended from those cells receiving the same type of building blocks that we still use today. And of course, that's applicable to all of the different taxa of organisms that use the same as well. So those are the ideas that I've seen with this particular idea. So just keep that in mind. And as I said, I'm going to definitely skip over this. If you guys are in AP Chem and aiming towards the end of the year, this should all be relatively simple. Just FYI, things like, radioactivity and fossils, fossil dating, carbon dating, not really that common on the AP exam. Okay. Um, let's move on a little bit to maybe this area here. Yeah, this is sort of the first thing that we can focus on because as we begin the review of Unit 1, we start with 1.1, which primarily deals with water. So I just want to briefly remind you guys the importance of polarity of water. Again, if you guys are in AP Chem, you have way better intuition as to what is happening with polarity of water. It really has to do with the electronegativity difference between the oxygen atom and the hydrogen atom. And this really changes the interactions between, well, at least the placement of electrons or the probability of finding electrons around each of these atoms. And of course, the more likely the electrons being found near the oxygen end is going to give the oxygen end a partial negative charge and then the partial positive charges on our hydrogen side. Now, one thing that I'll just quickly say here is that when you look at these charges in biology, just simplify it. Sometimes I have students who are in AP Chem and perhaps, in AP Physics as well, and they overcomplicate things in bio where it need not be that complicated. And I guess the main point that I'm trying to make is whenever you see negative and positive symbols in bio, it's about magnetic forces. Because you guys know electromagnetism is sort of a single phenomena. And the way that we think about this is not as much in terms of electric charges, but the way that they attract each other. So thinking about these as, the red side of a magnet and the blue side of a magnet, that's going to make things much easier as you deal with other interactions later on. Okay, so we're going to skip over a lot of this. But look at this one. Now, once again, this sort of goes into the realms of, I don't know, physiology to a certain degree, but I will say that this is a diagram that you probably are fully capable of understanding at this point, having done cell communications. On the left side, we have natural endorphins. So this would be sort of the feel-good neurotransmitter that kind of pats you on your back and says, "Hey, you're doing well." Or anyway, so I'm not a psychologist or psychiatrist, so I'm not 100% sure, but I know that it has to do with feeling good. And here we have morphine, which I presume most of you guys are aware that it's going to make you feel good. And the main point, again, of this picture is the structure and function. The idea that you have this little area, this little domain of the endorphin that's able to lock into endorphin receptors, and the fact that a plant-based morphine has the same type of structure here, regardless of what's happening anywhere else. That's what's allowing these molecules to actually unlock the receptors, so to say, triggering the same type of response. So once again, structure and function is coming into play. But as I said, we can take a look at 1.1 and go through this relatively quickly. So the first thing that we need to understand for water is just knowing how the properties can be explored within the context of the AP curriculum. And it all comes down to the hydrogen bonding. And I believe this picture is fairly accurate in stating that each water molecule does have the capacity to form four hydrogen bonds with water molecules nearby. Sorry, excuse me. I have allergies right now. It's, in Korea, we actually have the crazy seasonal changes right now, and the pollen drives me crazy. Anyway, so we have these water molecules that are interacting, and it really does give rise almost directly to these four different properties. Now, here's one thing that I want you guys to remember. Anytime something comes up as a set of four, those are the things that you really want to pay attention to. And I know that that sounds really silly, but hear me out, right? So basically, on this AP exam, the MCQ, you have four options, right? You have option A, B, and C, and D. And when you have something that comes as a set of four, what they can do is to give you three and remove one of them and have that last uh, one that's not part of the four be the answer for a negative question. So, for example, they might say, well, which one of these properties does not stem from the polarity of water? Now, when you answer that question, you have to know, okay, well, these are the four properties that we know that stem from polarity of water. This one is missing, and it's being replaced by something that is completely ridiculous because sometimes they do that. Well, that's sort of a strategy that AP Bio uses all the time. So sets of four as a test maker, right? Those are the things that make it really easy for us to test the students, fact-based questions, skill check questions. And here they are, right? Cohesive. And I'm going to actually real-time edit this right now. Let's also add adhesive, and I'm going to add surface tension as well, because these are all sort of collected together into a single rubric. And the main point here is not so much the chemistry of why and how these are happening, but the degree to which these play a role in biology. So, for example, we know that cohesion is the water's ability to be a little bit sticky because of all those hydrogen bonds. Adhesion, of course, is sort of water's ability to stick onto other things. If you take a little bit of water and put it on your finger, it's going to sort of defy gravity and stick to your hand. All of that's happening because, your skin is biological. It's also made from a lot of polar substrates, so water's going to stick to it. But biologically speaking, this is the main thing, right? I've seen this very picture come out on AP exams in the past. It might have been like a 2018, so it's been a little bit. But at the same time, they're asking in a MCQ, they show you this picture and say, well, which of the properties of water explains how transpiration works. And if you guys remember, transpiration is the translocation of water from the soil all the way to the top of the plant. And it's happening because of both adhesive and cohesive properties, where capillary action is allowing water molecules to literally climb on the side of the xylem. So xylem would be the water conducting cells, simultaneously pulling the water molecules beneath it. So, again, biologically relevant. And surface tension clearly can be important too, because it does offer a whole new ecosystem upon which organisms can survive. Now, Humans can't do this because we're just way too heavy, and the amount of force that's exerted by the surface area of our feet is not going to allow us to sort of stay above water. But when you look at not only insects like water striders, but dragonflies, mosquitoes that lay their eggs into the surface of water, as well as plants like lily pads that have leaves that are sort of adapted to floating on top of water. That is clearly going to be examples of how surface tension can be utilized by organisms to make additional ecosystems. Now, speaking of lily pads, by the way, this might have been almost a decade ago, but they had this one question, right? So the question was giving you the placement of the stomata on leaves of these unidentified plants. Now, if you guys remember what stomata are, these are the little holes that can be opened or closed for gas exchange on the leaves, usually on the bottom of the leaves, right? So that it can kind of stay in the shade and not draw out too much water, leading to drying out. But this one plant, which was an unidentified plant, had stomata on the top of its leaves. And the trick was that this was supposed to be like a lily, like a plant that had leaves that are floating on the surface. Because if those leaves were to have stomata at the bottom, then clearly that's not going to be able to exchange any gas because that's facing the bottom or that's facing the water surface. So I don't think the AP questions are as I don't know, I guess sort of tricky anymore, but that was definitely something memorable that I remember from one year. The next property of water is the moderation of temperature. There are a couple of things that arise here. First of all, there is the specific heat, but again, if you guys are in AP Chem, you know all about calorimetry. So thankfully, I don't have to explain this. If you do want a better explanation of this, I'm sure that there are many Khan Academy videos that go into a lot of questions that pertain to this. But for biology, what we need to know is just a simple fact, right? Water can absorb a lot of heat without changing its own temperature. Now, the way that we like to think about this is, well, there's a couple of things. For one, it really does allow water to absorb heat around coastal regions in order to sort of cool down the land area around it. But like for me, this is sort of like the worst biological example because Los Angeles is sort of the least natural thing that I've ever seen, right? So this is not really a good biological example. So first thing is to kind of demonstrate why it's so important that water has high specific heat. And it comes down to something like this. I mean, we can take a very ridiculous example. It's called reductio ad absurdum, right? Making a crazy example to demonstrate a point. So if we're to imagine that water had extremely low specific heat, such that just a little bit of heat would alter its temperature to a great degree, no pun intended, of course, and that would mean that every time the sun would come up, there would be sufficient heat to evaporate the entire oceans of our planet and put it into our atmosphere. And every night when the sun goes down, it cools down sufficiently that the water or the entire oceans that are now in the clouds rain back down, right? And every day this would happen. You'd have the water moving out of the ocean, water coming back to the ocean. Now, imagining that over the last 4.5 billion years, we've had ample time to evolve into everything that we see, in such a stochastic environment, in such an environment where just there's no stability, you're not going to get any organisms surviving. And that's why the stability of water is important because when you watch videos of like hydrothermal vents, there are these like lava that's spewing out of the ground in the ocean and is solidifying into rocks as it were, like in real time. And they're just like fish swimming by, just chilling. And they're not getting fried up because of the fact that all of that heat can be absorbed by water. So like for me, that seems like a much better biological example. And another thing is, of course, evaporative cooling, wherein the heat of vaporization of water, that would actually take the heat from the surface of the skin in order to cool you down. And this is not going to be applicable to every single species, but for humans, that's absolutely true. It's actually one of our major adaptations that allowed us to become basically who we are today. And of course, dogs. Like if you guys have ever seen dogs pant, that's what they're doing exactly. They're trying to use the only surface from which they can evaporate liquid, which is their tongues, to cool down. Now, another one is insulation. And this has to do with the fact that water, interestingly enough, actually gets less dense as it freezes as compared to when it's in a liquid form. And that means that, when a surface of a small body of water, and it doesn't even need to be that small, but When a surface of a body of water begins to freeze on account of its interactions with the cold temperatures on the atmosphere side of things, it will start to freeze. But if we're to again imagine that water actually becomes more dense, that ice would actually fall to the ground or to the bottom of that pond, allowing more water to freeze and then drop to the bottom. And effectively, it would just basically freeze bottom up. And that would kill everything within that body of water. But that's not actually what happens because we know that as water freezes, it actually forms a layer on top of that water. And that actually kind of like an igloo, right? So you can be in an igloo and still stay pretty warm if you're in the northern regions. And it's insulating that water underneath. And I think it's about like, 2 to 3 degrees underneath. And I know that that's not sufficient for us to survive, but for a lot of these, like shrimp species and so on, and even fish, they're perfectly happy as long as it's not frozen. And the last part, which is, I think, the most important, is the biological solvent, which is water. So water is typically thought of as the universal solvent. And here, again, because you guys are pretty comfortable with chemistry, I can show you a picture like this right away. And you'll know that when you have a crystalline structure of an ionic compound like sodium chloride, it could literally be broken down by water molecules on account of their negative and positive sides that are essentially going to break each of these ions apart. And essentially, when they do that, they sort of separate them into all of the individual ions now, randomly moving about with respect to just a kinetic molecular theory. And that's part of the reasons that it appears to be sort of disappearing, right? Because when we put a little bit of salt into water, given that we're not at a saturation point, it's going to seemingly disappear from our sight. And what's happening is not the actual disappearance, because that would be physically impossible, but just the fact that they're so far apart now, randomly moving, that they are not visible to us, right? The resolution wise, our eyes just simply can't see it. But this goes to a broader picture of describing what we call hydrophilic and hydrophobic substances, wherein we can definitely say that majority of the ionic substances are going to be hydrophilic as they dissolve in water, again, with the exception of certain ionic compounds that you guys know don't dissolve in water. But we also say, look, we can also add polar substance to this list. And when you guys were doing this in, let's say, Unit 1, you might not have really appreciated what this is saying here with the lysozyme, but lysozyme is an enzyme. And as you guys know, enzymes are proteins that are made from one or more polypeptides. Now, lysozyme happens to be made from a single polypeptide, so it's just got its tertiary structure. But you know that all of these amino acids can be categorized into different types of groups. So we have charged and we have polar and nonpolar. And essentially, the outer periphery of of this lysozyme has sufficient degree of polarity that it allows for the interaction of water, which means that even this big old enzyme can be perfectly happy in water. And it should be, because lysozyme is found in your saliva and mucus and so on, which is water-based. So that's a very important thing to note here. Okay. And that really does get us through most of water. I'm not including acids and bases here. Now, the typical textbook, does have that in this particular at this particular point. But there's a couple of reasons. For one, the only reason that you might need to know the differences between acids and bases is to correlate just how the pH corresponds to proton concentrations or H plus concentrations. And the only reason that really matters is when we talk about, let's say, mitochondrial inner membrane and the intermembrane space and where the protons are pumped to. That's sort of when where it links to it. I'm sure I'll talk more about that in our Unit 3 and 4 review. But for right now, don't worry about acids and bases too much. Okay, now when it comes to elements of life, this is sort of where we get to carbon chemistry. And once again, I think, AP Chem doesn't have orgo. I know that it doesn't have organic chemistry. So you don't have to know a ton about that for the time being, even for your AP Chem course. And you don't even need to know the naming conventions for these hydrocarbons. But what is important to note is that carbon is really great at forming all of these different varieties of structures. And once again, look, the overarching theme in the first unit is, look, if you want a lot of different functions for your biological molecules, you want to have a lot of different forms. And that's exactly what we see, because we can create a lot of different lengths of biological molecules. They can branch. And as you can see in this particular case, these are isomers, right? These are isomers because these are both C4H10, but they're shaped differently, and they should behave differently because they're shaped differently. So we have isomers. We have, of course, double bonding. We can have triple bonding even. And we have rings too, benzene rings and cyclohexane. Now, again, the important bits here is the idea of isomers, whether they're structural or geometric, um, and of course, enantiomers. Like I said, the most important bit is this diagram where they show you that a drug like ibuprofen, which by the way is Advil, if you have ever taken Advil, you have the left-handed Advil and you have the right-handed Advil. And the same thing with albuterol, which is that asthma inhaler. You have the right-handed albuterol and then the left-handed one. And you can see that the effective and ineffective enantiomers is not just about left side being always right or right side always being right. It all has to do with the idea that these drugs, once they enter your body, are going to engage with certain things inside your body, inside your cells, or on the surface of your cells. And again, I'm not a pharmacologist, so I don't know exactly the mechanisms of action, but what I can tell you as someone who teaches AP Bio is that there's high likelihood that each of these drugs in their right structure is going to interact with either a receptor or an enzyme within the cell in order to facilitate some sort of a cell reaction, right? And the only way that it can lock in to that particular enzyme or lock in with that particular receptor is by having the right shape. The example that I always give in class is, I know that a lot of people don't use keys anymore, right? Now everything is sort of either a touchpad or even a car, it's going to have push start button. But, when you imagine a traditional key and a lock, and you say, look, I'm going to take this key, I'm going to make a mirror image of it so that all the teeth that I have on the left side are going to go on the right side and vice versa. Now, you're not going to be able to open that lock because of the fact that the structure is intrinsically the opposite of what it was supposed to be. And again, that goes to sort of demonstrate that biology is not something that's happening at a quantum level. It's happening actually at a pretty molecular and a macro level, molecular level. And that's exactly why these things are not going to work either. Okay. All right. Now, when it comes to the functional groups, I think again, we're in a position at this point in March or April almost that we have the benefit of really appreciating where these things actually come up. So I'm going to do a little bit of jumping around here in order to demonstrate why they actually taught you all of these functional groups at the beginning of the year. We'll start with the hydroxyl group, which you guys might remember is this OH. I know that this might be a little bit small on the screen, but the hydroxyl group is super important because you guys know that whenever you look at a hydrocarbon, something like this, all of these hydrocarbons, they are all insoluble in water, as in they do not dissolve in water because they're all nonpolar. And in the context of a biology class, you have to wonder, why the heck am I learning all of these nonpolar substances if they don't dissolve in water, given that cells are mostly water and we all evolved in water? And the only way that we can actually fix that is by taking something like, let's say ethane, which is not dissolvable in water, and then just taking one of those hydrogens off and putting a hydroxyl group, as you can see here, turning it into ethanol, and now it is dissolvable in water, and it is biologically relevant because, well, that's just alcohol. Now, of course, alcohol in terms of our human race has played an important role. When you look at things like the development of Western cultures, they actually had to ferment a lot of their grains so that they don't go bad and it provided calorie and so on. So we have this polarity thing happening. And not only that, as I said, I'll jump up a little bit. Look at all these sugars, right? When you take a look at all these sugars, whether it be glyceraldehyde or glucose, all of these hydroxide groups that you see, well, they have been, previewed in that functional group to demonstrate that, look, all of these sugars are going to dissolve in water, and they should because, if they didn't, then how could we ever use them as an energy source? Now, what about the carbonyl group? Well, once again, the carbonyl group seems a little bit weird. It's just this double bonded oxygen. But if we go back here, see, you can see that's where the carbonyl group is, or that's where the carbonyl group is. And that's one of the distinctions that we make, because you might remember aldehydes have the carbonyl group at the end of that carbon chain. Ketones have it in the middle. And that's one of the reasons that we actually differentiate and discern these types of sugars, although I don't think that's as important for AP Bio. What is important for AP Bio are the next two groups, which is carboxyl as well as the amino groups. And that's because the carboxyl and the amino groups make their appearance in an extremely important substance that we call amino acids. So important because we see these time and time again. I mean, I would say protein structure and function forms at least half of what the FRQ sections are going to be. So maybe that's not true. I would say maybe 30%, which is a pretty big portion. And here you see the amino group and the carboxyl group, and that's precisely why they're giving you this preview of the carboxyl amino group. Sulfhydryl group. Does anyone know where the sulfhydryl group comes out? This is actually pretty important too. You can type it in the chat if you remember. Sulfhydryl group. So we're going to go back to the amino acids. And let's go to cysteine right there. That's the sulfhydryl group. That's the only place that we really see the sulfhydryl group come out in the course of AP Bio. However, even though it only comes out here, there are two reasons that the sulfhydryl group is so important. For one, I don't know if you guys remember something called the Hershey-Chase experiment. It was an experiment that attempted to figure out whether it was protein or DNA that formed the genetic material. And what they did was they radioactively labeled sulfur and phosphate in bacteriophage growth or growth plates, and then they infected bacteria with these radioactively labeled bacteriophages, and they discovered that what actually went into the bacteria was phosphate, indicating that that was DNA. And the reason is that DNA has, of course, carbon, hydrogen, oxygen, and nitrogen, but it does not have sulfur, but rather a phosphate. Whereas in proteins, it has, again, carbon, hydrogen, oxygen, and nitrogen, but it does not have phosphate, but rather sulfur. So it really does play an important role in some of those, historical experiments that leveraged the presence of sulfur. And of course, sulfur also comes out in methionine, by the way, but it's not a sulfhydryl group. But it was this distinction that made a really big difference in our discovery of DNA as a genetic material. And the other thing is that of all the R group interactions that we talk about in the folding of polypeptides, there's really only one very, very strong interaction, which is covalent bonding. Let me skip ahead right here. So you have things like van der Waals forces, you have hydrogen bonding, which is a weak, intermolecular force. Ionic bond's a little bit better, but covalent bond, as we know, is going to be pretty strong. And the only way that covalent bonds form in our tertiary structure is by having these disulfide bridges that can actually create this covalent bond as they lose the two hydrogen atoms. And in fact, it's so interesting that we actually see those sulfur disulfide bridges here, even in a diagram like this. So, I don't know the remainder of what the amino acids are, but I can tell you that on this side and on this side, we have cysteines. On this side, we have cysteines, right? So you can kind of see how important that sulfur or sulfhydryl group can be in our investigation of polypeptides. Now, let's move on a little bit. Phosphates. Okay, so I think at this point, you guys are pretty familiar with where these phosphates come in. ATP, right? Adenosine triphosphate. We also have the phosphate sugar backbone in DNA. And we also see phosphates on phospholipids, right? So the lipids that form our cell membranes. So I think it goes without saying that phosphates have already claimed its throne as one of the most important functional groups. And then the last one is the methyl group. Now, I'm not going to go into too much detail about the methyl group, but I will say a couple of things. And it pertains to the things that you need to know. Firstly, as I mentioned earlier, hydrocarbons are not interesting biologically. I said that they don't dissolve in water. They're not going to be very interactive with biological substrate. And you can kind of see that the methyl group is sort of like that, right? It's basically this little portion here with the CH3 that we can add onto something. But what's interesting is that because these hydrocarbons are not that biologically relevant, when you add methyl groups to, let's say, a base, you can actually make that base less reactive to the things that typically interact with that base. So one prime example of this is by adding these methyl groups to a sequence of DNA, it could actually effectively silence that gene. Because once it has the methyl group and has an altered structure that makes it less biologically reactive, it's not going to react with things like transcription factors. It might not bind to, let's say, RNA polymerases properly. And the idea is that when we talk about developmental biology and we say, look, there are cell differentiation events happening where certain cells are not going to be producing this set of genes because it doesn't need to, well, methylation is one way of doing it. And the other thing that I'll mention, and this is not going to come out till about Unit 6 when you guys do, molecular biology, but you guys all remember 5' caps, yes? So the purpose of five prime caps were threefold. One was to facilitate its export, the export of mRNA from the nucleus to the cytoplasm. The second was to facilitate the binding of the small ribosomal subunit. But the most important, and I would argue the main reason for the existence of the five prime cap was to inhibit nucleases from breaking down that mRNA once it's in the nucleus. And the way that it actually did that is by adding a five prime cap. But what is a five prime cap? Well, it happens to be a guanosine triphosphate cap that's been methylated. It's actually a methylated guanosine, and it's a triphosphate, so it's attached to the five prime. And again, because of that methylation, it makes it less likely for it to interact with enzymes. And again, it sort of stems all the way back to what we're learning here in Unit 1. All right, great. So that gets us through majority of the background material. Let's have a little bit of tea here. I just realized that I had that slide backwards, but how are you guys doing so far? Any questions, by the way? You guys can always use the raise your hand function or just type in the chat real time. And because we're recording, I don't know if the recording function captures the chats. I'll read it out loud if you guys have any specific questions. But all right, let's move on then. Macromolecules. So there are a few things to say about macromolecules, obviously. Actually, way more than a few things. But, whenever we talk about macromolecules, we start by defining a polymer and what monomers are. And I guess I've seen questions like this come out where they ask about, you know, just this very reaction. So let me see if I can actually find it. Okay. So as we go through this, just give me one moment. I think it was actually this one. Okay. I know I don't have the question on the slide right now, but okay. All right. There we go. All right. So we're looking at the slide here. And as you guys know, there are two different reactions that you need to be familiar with, one of which is dehydration reaction. This is also called condensation reaction or dehydration synthesis. They all mean the same thing. Essentially, you have the monomer being linked to a polymer or another monomer. And the idea is that with the removal of water, you're able to sort of form that covalent bond. Okay, so this should be H2O. There you go. Um, and on the other hand, we have hydrolysis, which comes from the word hydro, meaning water, and lysis, meaning to cut. So to cut with water, you would basically be doing an inverse reaction. Now, I'm sure if you guys have done some previous year FRQ, you may have seen some math questions where they say, look, I have 200, 200 amino acids. How many nucleotide bases does it take to encode for that, plus the stop. So you'd have to like do 200 times 3 because each amino acid is encoded by 3 bases and so on. There is a math question that could potentially come out, and it really is just this idea of how many water molecules would you form or how many water molecules would you need in order to break something. It's very, very simple. The idea is always n minus 1, right? So if you guys, I mean, some of you guys are in AP Calc or finish AP Calc from what I read, clearly when you're doing the mathematical derivations like this, when you have 4, you need 3 water molecules, or you form 3 water molecules. When you have 5, you form 4, and so on. So just be aware that those types of like really silly, simple math questions can come up because, sophomores or even sometimes freshmen take AP Bio, so they still talk about that as well. But as I said, there is some relevance to this slide, and there are two things that I want to say. First of all, one of the main things that I want you guys to remember is that in Unit 6, we talk about protein synthesis where in the ribosomes, you have the polymerization of polypeptides by, each amino acid being added together, right, in the P site and so on. That is exactly what's happening in this dehydration reaction. So like, even though, this isn't so clear that these are amino acids, it's a model that we do go into more specifics later. And the other thing that I want to mention is that you guys have to understand that the polymerization process, so creating complexity from simpler beginnings, is going to be something that reduces the entropy of that particular molecule and requires energy to do it, right? Because in Unit 3, when we talk about energy, we learn that when you go from simple to complex, you always need energy. And here, even though in Unit 1, they're not showing you the transfer of energy, we have to understand that what we're seeing here in figure 5-2A is an endergonic reaction. And what we're seeing in 5-2b is an exergonic reaction. And the breakdown through hydrolysis should theoretically release energy, even though it's not being shown here. So again, there's a little bit of information that they are excluding because you haven't gotten to those chapters yet. But as I said, we have the privilege of sitting here in March so that we can actually add those extra details to a simpler model like this. Now, the last thing that I wanted to show you guys is this question. This is from 2024 AP Bio. So this is last year. Now, from what I understand, most of you guys are not in the United States. So these are going to be, I guess, American domestic questions and international questions are a little bit different. They're not different in format, they're just different in context. So it's not going to be that different. But here, take a look at this. Describe the role of water in the hydrolysis of ATP. And once again, they're not asking for, like an organic chemistry answer, right? Like, oh, it's going to attack it from this angle. No, no one expects you to do that. All they want you to say is, look, whenever you break things down in biology, whenever you're breaking covalent bonds, which that's exactly how each of the phosphates are attached to one another, we use water. And in this hydrolysis reaction, water is used to cleave the bonds between the ATP or the phosphates in ATP to create ADP and phosphate. And that's it. That's all you have to do. And that's like 1 point. And I know it's just 1 point, but every point counts on FRQ, right? You want to get as many points as possible, especially on an easy question like this. It's stemming right from what we're looking at in this very slide here. Okay. All right, so carbohydrates. I don't know if I'm gonna go into like all of the nitty-gritty details about these, but remember that there are a few things that you want to know. First is function of each of these macromolecules. So carbohydrates mostly going to be energy source, but also structure in terms of cellulose. You also want to know what they're called generally. For example, monosaccharides is what we call the monomers of sugars. Polysaccharides is what we call polymers of sugars. As far as this is concerned, again, I think there is something interesting to be said about this particular slide because in the very first unit, we primarily focus on glucose. And most teachers will say, look, let's just worry about glucose. But all of these come out throughout the course. And I'll hopefully be able to trigger some memory here. First of all, glucose and fructose combine together into a disaccharide that we call sucrose. Now, we learn a little bit about sucrose because one of the enzymes that they teach you about in chapter 8 or the enzymatics is sucrase, which has the capacity to break down glucose and fructose. All right, so that's fine. Glucose and galactose makes something called lactose. And that's important because lactose is essentially that sugar that triggers the lac operon when we talk about gene expression regulation in bacteria. So we have like beta galactosidase or however you pronounce it, that's going to be breaking down lactose. Ribose sugars are found in RNA. Deoxyribose sugars are found in DNA. Glyceraldehyde 3-phosphates are found within our Calvin cycle as well as glycolysis. Dihydroxyacetone found in glycolysis as one of the major substrates that we get, which gets converted into glyceraldehyde, by the way. Ribulose, remember ribulose 1,5-bisphosphate? That's going to be the very beginning of the Calvin cycle where Rubisco adds carbon dioxide to ribulose 1,5-bisphosphate. Now, it just happens to have a phosphate on either ends, but this is the base sugar. So essentially, everything that you see here does make an appearance later. But again, in Unit 1, it's almost like a character list. If you're reading Shakespeare, which, by the way, I hate. I'm totally a STEM guy, but whenever you read Shakespeare and you're reading Macbeth, they have this character list. It's like, oh, Macbeth is the. See, this is how much I know. I almost said Prince of Denmark. I think that was Hamlet. Anyway, the idea is that they give you all of these character lists and what they're supposed to do. It's sort of what you're seeing here, but they do make much more sense when you go back and appreciate it a little bit later on. Glycosidic linkages are the covalent bonds that we call it in sugars, so that here's where we see the maltose and sucrose. We don't see galactose as a diagram for some reason. But remember, polysaccharides are important. And here we have starch and glycogen. These are both storage polysaccharides. Now, one of the questions that I'm often asked is why do plants store them in starch? And the best answer to this is that, having every glucose molecule in its glucose form actually makes it more reactive and more likely to break down under, let's say, glycolysis and cell respiration. And that's not always a good thing because remember that cell respiratory processes, and this is, by the way, really important. I'm going to type this in, right? So cell respiration, like any energy conversion process, produces heat. Okay, heat, not hate. Sorry, heat. And the idea is if you have all this glucose that are just freely roaming around and you break them down, you produce too much heat. That's not good for the plant, right? So you actually want to keep it in there. Now the analogy to this from real life is like walking around with a bunch of cash in your pocket. Now I know that in 2025, having cash is actually worse than having a credit card, and I know that because that's just the way it is. But before when I was growing up, when I was a kid growing up, the more cash that you carry in your pocket, the more likely you're going to spend it, right? So you put it in a piggy bank, or you actually put it in an actual bank, and then you can have it when there's a rainy day, and that's sort of the similar situation for glycogen, except for animals. Obviously, glycogen is going to be a little bit more dynamic, as in like, okay, let me explain that quickly. So when you see starch molecules in plants, they're mostly in things like potatoes, like tubers, or some permanent storage because they make so much of it and they can put it away and not worry about it for a while. But for animals, we don't have this indefinite ability to become larger and larger and larger. I mean, at least like, I mean, you can get fat, but like that's not the same as a plant just continuously growing vegetatively and propagating vegetatively. So in terms of animals, we regulate a certain amount of blood sugar concentration, and we do that with insulin and glucagon, which hopefully you guys will study or have studied. So any extra sugar that's roaming around in your bloodstream is going to be converted into glycogen, but because we also do not eat constantly. And right now in Ramadan, you guys are doing fasting. So you know this better than anyone else that in times where you're actually not intaking any carbohydrates, that glycogen that you've been storing in your liver is going to be released back into your bloodstream. And that's precisely how the dynamics of this is going to work within a single day or within a single hour. Whereas with plants, it's a little bit more of a permanent thing. But what's important about this? Well, what's important is its comparison. My mic being weird? It seems like the volume is coming in and out, at least in my monitors. So I'm going to try to be a little bit more careful here. Um, what's important is how this actually interacts with the idea of cellulose, because cellulose is again a plant polysaccharide, but in this case, it's a structural polysaccharide. Now, there is this idea of alpha and beta glucose. It's rare that they ask about this, but again, if they were to ask about any differences between alpha and beta linkages, it all has to do with the fact that they're structured differently. And the enzymes that break down, let's say, starch, which includes, your amylase or any other, carbohydrates that we learn about, they're not going to be able to recognize the bonds that we see in beta linkages because, again, enzymes and their substrates operate based on structural um, correspondence. And in this case, that's the reason that cellulose is indigestible by animals because of the fact that we do not have any enzymes that can naturally break down cellulose. However, the idea that we can sort of extend this onwards into even Unit 8, where you talk about symbiosis and mutualism, is that herbivores who primarily rely on this type of, let's say, leafy green substances, they have bacterial cells that, again, because they're not animals, bacteria are not animals, they have cellulase. That's the enzyme that can break down cellulose. And as a result, they're able to sort of, provide sugars to the cow, and the cow in return would provide, a warm place where they can propagate. And just keep in mind that there's also another type of organism that is often invoked within the AP curriculum, and that is hold on. I want to say it's a protozoan. Okay, so it'll be under the kingdom Protista, which is sort of like the dumping bin, right, of classification because they're all sort of paraphyletic. But the idea is a lot of protozoans do have the capacity to break down cellulose, and those are the ones that would be interacting with termites. So again, let me just briefly talk about that for a moment because here in Korea, I am from the United States and of course Canada, but um, we don't see termites in other parts of the world as much. So again, if you're not in the United States, there may be some cultural issues when you're taking this test. But termites are these they're not like ants, but they kind of look like ants and they eat wood, right? They eat the actual woody tissue. And in America, because most houses are built with wood, whenever you're doing a home inspection before buying a house, like the first thing that you do is to check for termite damage, because if there's termite damage, it's gonna cost you a lot of money, or you might even have to rebuild the house. So the question is, how do termites do it? Because termites are animals. I know that we don't typically think of arthropods and things like that as being animals, but they are certainly animals. Again, it's the salivary glands of termites that house these protozoans, and we've seen questions that pertain to that very specific idea in the past. All right, quickly moving on to lipids. Now, lipids are not true polymers. That is to say that they actually don't indefinitely get longer and longer and longer like glucose polymers can get, or polysaccharides can get. We learn about three different kinds. The first is fats and oils, which are basically glycerols combined with three fatty acid chains. These are going to be basically long-term storage of energy for most organisms. There is this idea of saturated versus unsaturated fat, which I don't think is as important for AP Bio, unless we're talking about this in the context of membrane fluidity, which we'll talk about in the second half. So we'll quickly go over this. Phosphate makes an appearance here. The most important part about phospholipids, obviously, is the fact that they are amphipathic. And amphipathic means that they are at once hydrophilic and hydrophobic. And when you add them into water, or at least a bunch of them, you do get this wonderful, what we call phospholipid bilayer. And, again, the next section is going to be primarily focused on these membranes, but, it's going to be almost like the perfect thing that you can, dream of in making a membranous structure. Here we have some steroids. Steroids are, well, they're not going to be explored in as much detail, except for the fact that you just have to remember that steroids can be hormones, and these hormones, as they're lipids, are going to be lipid soluble. And as a result, when we study things like cell communication in chapter 11, we learn that these are the signaling molecules that have receptors on the inside of the cell. They have intracellular receptors because they don't have to bind to the receptors that are facing the outside. They can go straight through the membrane, no problem, right? So those are some of the things that you might want to remember. Okay. So the next big one is proteins. And here, let me make a couple of points. So we see a very, very interesting table here, table 5.1. Okay, now remember, this 5 means this is chapter 5. And they tell you, look, these are all the things that proteins do. Enzymatic, which, by the way, comes out in chapter 8. Structural, all of these things, you don't really see them in this textbook. Storage, transport, hemoglobin. Okay, you might see it a little bit. Hormonal, receptors. Okay, that's Chapter 8, contractile motor protein. That's from physiology. Defensive, that's, of course, from the immune system. Look, the book knows that you don't know all of these things, right? That you haven't talked about any of these yet in the course. But the only reason that they actually give you this table, and again, I don't want to sound like a broken record, but here's the thing. They're telling you that, look at all of these different functions that proteins have. And that means you're expecting to see a lot of structural diversity in proteins too. That's basically what they're saying. So proteins can do all of these different things because they can come in all of these different shapes and sizes. That's what they're trying to prove here. So the question then is, well, how? And the answer is this, the 20 amino acids. Now, I know that a lot of students get freaked out. They say, do I have to memorize all this? Well, I mean, if you have a very sadistic teacher at your school, maybe, but for the AP exam, not so much, right? For the AP exam, you do need to know how to sort of interpret the chemical qualities of each of these groups, but not necessarily very specifically. Let me give you a couple of examples. So for one, you should know that these are nonpolar because these are all hydrocarbons, and we learn hydrocarbons are boring, right? We said that they don't react with biological, or at least water. So these are going to be nonpolar. Hydroxyl groups we know are polar. Amino groups we know are polar, right? Or ammonia we saw as one of the major examples of hydrogen bonding. And then when we have charged, like acids and bases, well, acids are negatively charged because they added the proton into the solution. Bases are positively charged because they obtained the proton from the solution. And these are clearly going to be water soluble because, as we saw with sodium chloride, these are charged, right? So all of these ideas all go together. But what's important is that because you have all of these different types of amino acids, when we combine them together into a long chain, there are local interactions and distant interactions that creates a three-dimensional shape, which is what we call the conformation of a protein, which is kind of a good name because it conforms to the environment in which these amino acids are laid out. So, for example, because these are usually in cell environments or cytoplasmic water-based environments, you're going to see a lot of these hydrophobic groups that are facing on the inside, hydrophilic regions facing on the outer domains. Of course, you have disulfide bridges, ionic bonds forming from bases and acids, and all of these things are happening naturally. And what's fascinating is that this entire idea of the sequence of amino acids that we learn in Unit 1, as you'll find out, is dictated by the sequence of DNA that we have in each one of those genes. And because sequence is what results in the three-dimensional shape, and the three-dimensional shape is what results in the function, you can actually draw this straight arrow between DNA code and the function that a form is that a cell is performing, indicating this really nice idea that DNA is really the blueprint of life because it dictates how a cell ought to function and what it's going to do. And it really all does make sense from the bottom up, right? And that's one of the nice things about biology that we have this connectivity. Now, when it comes to the details of things like what kind of bonds form secondary structures, it's nice to know that these are hydrogen bonding between the backbones, but I don't think it's absolutely necessary. But what is important at this point for me is to say one particular thing. So, for example, when you take a look at um, yeah, this slide, you see this lysine and arginine. And this is something that's come up at least twice on previous AP exams. So I'm telling you guys for this right now. And I don't think, this is illegal. Like, I'm not showing you guys the questions, but I'm just kind of giving you guys a general idea. So when you look at a question and it deals with primate phylogeny, right? For example, we're looking at things like humans, chimpanzees, and gorillas, and so on. What we see is that on our hemoglobin protein, gorilla lineage has um, lysine instead of arginine in one of its positions. Okay, I forgot which position it is. It's like 142nd position or whatever. But so chimpanzees and humans have arginine in that particular position of the hemoglobin beta subunit, whereas the gorillas have lysine. And if we go farther out and look at some other mammals like horses, they also have arginine. So we know that this is arginine is sort of like the conserved trait, right? Now, in the context of what we're discussing here, I remember, in a couple of instances where they ask, well, how is it that gorillas are doing just fine? How is it that they have a mutation that took this arginine and converted it into lysine or vice versa? And how is it that they're doing okay? And the main answer to that is that because these two amino acids are in the same category of amino acids in terms of their chemical structure and their chemical function, when they're placed into a polypeptide, let's take a look at something like this. When they're placed into this polypeptide, I mean, we can even zoom in on that lysine right there. So let me just zoom in and let's say, look, let's say that we take this lysine and convert that into arginine. The question is, would that have as great of an impact on the overall structure of that polypeptide? And the answer is probably not, because the chemical interactions are good enough, right? And it's almost like, I don't have a hammer, but I might have, something that looks like a hammer, and I can use it to put a nail in the board, right? It's good enough. And that's essentially what we call a conserved missense mutation, right? A missense mutation is when an amino acid switches, but it's conserved as in that it's still using the same kind of amino acid, it's still within the base, and that is how you would sort of address that type of question because it all contributes to the overall structure of a polypeptide. By the way, for hemoglobin, here it is right there, the beta subunit, right? They love this hemoglobin because we also see it in things like sickle cell anemia. Now, in the case of sickle cell anemia, take a look at this. The sixth position, we go from glutamic acid, which from our discussion thus far, you would notice this would be a negative, right? It's an acid, so it's a negatively charged amino acid. It's definitely going to be water soluble. It converts into valine, which, without going back to our original picture of the amino acid chart, this is a non-polar amino acid. So now you have a You have a non-conserved missense mutation, and things just are not going to happen well here. Things are not going to go well, and that's precisely how we get sickle cell anemia, and that's what we see in this picture as well. A little bit about denaturation, although denaturation ideas are far more greatly explored in chapter 8. By the way, this is absolutely important. Like I guarantee it. I guarantee it, guys. You're gonna have to probably. I say guarantee and probably, but I'm trying to cover my butt here. Look, you're likely gonna have to explain why pH or temperature can affect the optimality of a protein function. And the way that you do this is by exploring all the way back to unit 1 and saying, look, proteins are held together, or the structure of polypeptides are held together through chemical interactions. All right. When you have too high heat, those chemical interactions become compromised because of the fact that the bonds become, well, weaker and weaker as the molecules vibrate faster. And as those bonds break, the structure of the polypeptide breaks down. And when the structure of the polypeptide breaks down, the function of the polypeptide breaks down. You see how it all still connects to the same idea. You will have to just basically regurgitate what I just said, for those full points on explaining why proteins outside of their optimal range is going to have those issues. Okay, great. Now, DNA, I am not really going to focus too much on this today because of the fact that we review all of this content when it gets time to studying DNA gene expression in Unit 6. So we'll put a pause on that, which is nice because it's about an hour in at this point, and we're kind of halfway through. Although I would argue that we're more than halfway through because when it comes to cell structure, function, and size, this is a much shorter review. So I suspect that this is going to take about maybe half an hour or so, which will still give us a little bit of time. And I don't mind going a little bit over 2 hours today if we need to. But let's just actually pause here for a moment so we can just quickly get some more coffee, make a bathroom break if you guys need to do. And then we'll start again in about 10 minutes past the hour. All right? So let's pause here for now. Okay, so we're back. For those of you guys who are still here, I think we did lose uh, one person. Hold on. Let's see if anything's in the chat. Nah, it might be getting a little bit too late. I think in the Middle East it's probably around 3 a.m. at this point. Um, yeah, it's getting pretty late. I'll try to go through this the remainder of the segment a little bit more quickly for you guys, so you guys can get to bed. And next time, here's what I'm thinking. Next time, maybe I'll just do this on YouTube Live, and that means that anyone who's around YouTube, wherever they are, can find us. And of course, it will probably be recorded and then just put up on my channel automatically. So I think that might work out as well. And this is great because yeah, for me, it's like my dream of being a StarCraft live streamer never panned out because it turns out I was better at biology than StarCraft. So I still have the whole setup here. But anyway, let's continue. So the second part of I don't know why I have this as 2. I think this is oh yeah, this is unit 2. That's right. So moving on to the second unit, we deal with cells. And here's one thing that I'll say about cells. Okay, so there are some larger ideas that you definitely want to know that connect to things like, everything's made of cells because the universality of living organisms and so on. But there are some major themes that I want to focus on here. And we'll begin with the idea of cell sizes. Now, microscopy stuff, this is important for certain courses, whether you're in IB or AP, your teachers might ask you to calculate magnification and things like that, but that never comes out on the AP exam. But what is important is understanding why the cells are the way that they are in terms of their sizes. So, a question that I always pose, which seems like a really silly question, is to ask yourselves, why is it that hamsters have fewer cells than elephants, as opposed to elephants just, having bigger cells? And, let's say every animal is supposed to have 100 cells. I mean, it sounds like such a ridiculous question, but really it gets at the heart of the issue where we say, well, all cells have to remain at a relatively small size. And I hope by this point in the review that you guys remember that the surface area to volume ratio, not the absolute surface area, right, but the surface area to volume ratio decreases as any object gets bigger and bigger and bigger. And that is to say that whether we're looking at a square or cube or a sphere like a cell, as it gets bigger, you're going to lose that surface area. And the main idea as to why that matters has to do with the fact that the membrane that we'll talk about in more detail in a bit is the surface area through which either enzymatic reactions can occur, or better yet, you have exchange of materials like oxygen coming in and carbon dioxide going out for aerobic species, and it's all going to be, let's say, relevant to the volume that it has to sustain. So, for example, as the volume gets bigger, the presumable fact here is that you're going to have way more oxygen and you're going to produce way more CO2, and you're going to have to commensurately release that CO2 and get that O2 in. But, if the surface area continues to get smaller and smaller and smaller, relative to that volume, at some point you're going to get choked out. It's kind of like trying to breathe through a little Starbucks straw, the coffee stirring straw. You're just not going to be able to get enough air in. And that's sort of the same thing that would happen to cells. So they eventually end up partitioning into smaller and smaller and smaller pieces. Now, there are, of course, exceptions to the rule. Some of the nerve cells can get quite big, but that's primarily because their structure, the shape itself is just kind of elongated, and they still do have relatively large surface area when you compare to other cells of the similar size. Um, all right, now going back to some of the major differences between the types of cells that we have. Look, there's a bigger picture to be said here, right? So to be analyzed here. So even though here we're looking at, a bacterial cell, and we can talk about how, yeah, they don't have membrane-bound organelles and all of that. The main thing that I would argue is maybe going to my browser and giving you guys a different picture. So let's say we go to something like, let's say, Tree of Life biology. Okay. All right. So when we take a look at something like the Tree of Life, this is a pretty good example of it. What we see here is this green line, and this is the lineage in which eukaryotic cells evolved. So that's kind of a better thing to think about, right? So at this point in the branch that we see as green, that's where things like membrane-bound organelles would evolve. That's where we have things like the nucleus evolving, and of course, the very important idea of the migration of aerobic bacteria like mitochondria's predecessor, forming that endosymbiosis, right? That's what's happening. But another way to think about this is, well, then what is it that connects everything together, right down to the last universal common ancestor? And that's sort of where the analysis of the cell actually begins, because they say, well, all right, so I know that there are distinctions between these different types of cells, but the basic features of all cells is that we have the plasma membrane that is encapsulating the cytoplasm. And of course, everything is going to have DNA, and it's going to be capable of expressing the genes on that DNA through the gene expression pathway involving ribosomes and hopefully messenger RNA that's somewhere in between. Um, Again, we have a set of four. Now, once again, you want to be really familiar with being able to identify three features that are similar between prokaryotic and eukaryotic cells and one that's not. Or alternatively, you want to be able to determine what three features are different, but one that is shared. Those types of questions arise a lot on these MCQ. Okay, so that's what you want to pay attention to. Now, one of the things that we see in this chapter, this, by the way, I always like to think about this in terms of chapters because Campbell biology is such a great basis for this exam, um, is that in this chapter 6, they do actually go into some information about every single one of these organelles. But for the purpose of the AP exam, um, it's not so important that you understand everything at this point because of the fact that for each one of these things, like mitochondria, we have a whole chapter 9 devoted to the function of the mitochondria and its cell respiratory pathway. We have entire chapters devoted to what's happening inside the nucleus. So once again, it's kind of like that, character list at the beginning of a play where they tell you, well, this is what it does and that's what that does. But, we're going to explore it in much greater detail because, again, this is only chapter 6. So you don't have to be so worried about the details when it comes to this chapter. However, if there's one thing that I would really emphasize is this picture right here, because this picture demonstrates something called the endomembrane system that you do have to take away from this chapter. And the endomembrane system is basically a collection of membranous organelles, starting from the nucleus, which is actually contiguous with the rough ER and the smooth ER. And then it also connects to the Golgi body as well as the plasma membrane. and even lysosomes. And it's happening through a continuous flow of plasma membrane exchange via the vesicles or the vesicles or the vesicles, depending on how you want to say it. Look, the main point is that any plasma membrane component, like a phospholipid found on the plasma membrane, could at some point be found back in the ER and vice versa. Oh, okay. So hold on. We have someone coming back. Let's see if that's going to work out. I have to wait. Where's my Zoom controls? Ah, there it is. Ah, there we go. All right. So the endomembrane system, as it were, is the continuation of the membrane through the vesicle structures or the vesicle structures. And and the main thing to note is that as we talk about protein synthesis, we need to understand that proteins that are either destined to be released, as you can see in this image, is going to be inevitably formed into the endoplasmic reticular lumen, specifically the rough ER, because that's where the ribosomes are. And once again, when I teach this in the beginning of the year, it's kind of hard for students to understand because they haven't really learned, I mean, unless they've done a general biology course, they haven't really learned a lot about ribosomes and what it does. But, once you have done Unit 6, you know that the ribosomes are responsible for producing proteins. And when they do that on the surface of our ER, they're able to place that polypeptide through the translocation channel into the ER, where they can be packaged into vesicles, and then they can be modified into Golgi and then eventually released. And of course, this would also apply to membrane bound proteins. So we're talking about things like receptor proteins or proteins involved in the electron transport chain. Well, not necessarily on the surface of our membranes, but proteins like that, transmembrane proteins, are all going to be still placed into the ER surface and then packaged elsewhere. Now, another thing to note, and this was once again something that I remember from a previous exam, is that they talk about, hold on, let me try to get this right. They talk about um, yeah, they talk about the formation of lysosomes. Now, you might remember that lysosomes are membrane-bound organelles that have hydrolytic enzymes, right? Again, hydrolysis, right? The breakdown of covalent bonds. So you have these hydrolytic enzymes in lysosomes that are supposed to kind of act like um, the garbage disposal, right? This is sort of where all the breakdown occurs. But the idea is that if you have some problem of lysosome formation, right? So the last part where the lysosome forms here in this picture, if you have a problem with that, then where would all those hydrolytic enzymes sort of get stuck at? And the main answer in that case was the Golgi, because remember that these enzymes are initially produced into the ER. They're presumably modified in our Golgi apparatus, but because they cannot come out to form the final lysosome product, they're going to get stuck in the Golgi apparatus. So like this type of migration of material is another thing that you want to keep in mind. And on the AP classroom, for those of you guys who have had an opportunity to solve questions there, you may have seen questions where they ask about, well, what about the mRNA? Like if you're to radioactively label mRNA, where would you find them in this entire process of protein export? Well, the mRNA would still be found near the ER, right? They don't go into the ER. The mRNA snakes out through the nuclear pore and engages with the ribosome, but ribosomes are just stuck on the surface of the ER, so they wouldn't be going anywhere. And that's sort of to distinguish the path of the proteins versus the path of the messenger RNA within the gene expression pathway. All right, now, I mentioned this earlier, but a very important thing to note here, and guys, this is again going to come out on the test. Almost every year, they have at least one question that pertains to this idea, and that has to do with the endosymbiosis theory. So the idea that things like mitochondria as well as chloroplasts were at once a free living species of prokaryotes that have since moved in and kind of created this most intimate symbiotic relationship with eukaryotic cells. And they usually want to know at least two pieces of evidence that supports that theory. Now, one piece of that evidence is, of course, the double membrane. And the double membrane is really in reference to the idea of endocytosis. If you guys remember, endocytosis is this bulk transport where, and I think I could probably reference a picture here somewhere. Oh, well, turns out I don't have the picture of it. Oh, actually, I do. It's in the next chapter. Hold on. Yeah, it's this sort of idea. But you can see that if you were to perform endocytosis for a bacterial cell, you'd have this bacterial cell membrane and the membrane of the engulfing cell that surrounds the outside. So there's that first piece of evidence that, yes, perhaps we have a symbiosis event. And then the second one is that mitochondria actually have their own DNA, right? Mitochondrial DNA as well as chloroplast DNA, they're real things, and this does actually link up with some of the ideas later on when we do genetics. I know that some of you guys, I don't know who it was, but was lamenting about Unit 5 and how difficult it was. But in that Unit 5 in genetics, we learned that mitochondrial inheritance patterns are also things to look out for, which means that clearly there's DNA there. And of course, if I were to add a little bit more detail to that, the DNA that's actually found within mitochondria and chloroplasts are circular chromosomes. And you guys might remember that circular chromosomes are very, very emblematic of prokaryotic DNA structure. And lastly, they actually have their own uh, prokaryotic ribosomes, which are, by the way, a little bit smaller than eukaryotic ribosomes that are operating within each of these organelles too. And they dictate their own division and so on. So there's a ton of evidence that they are, prokaryotes that are actually just, making themselves at home within eukaryotic cells. And that much is, undoubtedly true at this point. If anything could be true in science, I would say that that's got a very high probability of being true. All right. Now, that's basically it. I mean, there are these things like microtubules, filaments, and intermediate filaments. I will just say a couple of things here that I really like talking about, particularly because I have some really interesting experience with some plants that deal with this idea. Hold on. Yeah, I should probably explain that a little bit further. Look, microtubules have this thing called dynamic instability. These guys can sort of polymerize on both ends, which make it seem like they're actually getting like longer. They can depolymerize on both ends, making it look like it's getting smaller. They can actually polymerize on one end and depolymerize on the other, and it almost looks like it's moving through the cell. So they can do a lot of this. And the reason that microtubules are so important for biology, at least for AP biology, is that they are the spindle fibers that are involved in the chromosomal movements during cell division. And the plant thing that I was talking about is that one year I was up in northern Canada, and we were collecting plants, and you know a friend of mine, he had this plant, and we were just joking around. And I don't know why, but like I was gonna like eat it, and we're daring each other and stuff because we found like gold thread, which is like super bitter, so we're daring each other to eat it. And then I almost ate this plant where my friend was like, "Oh no, dude, you can't eat that," and that was because it was actually a yew tree, right? And I don't even know if it was naturally found or if he brought that in, but a Pacific yew tree contains a particular chemical called Taxol, and Taxol has this ability to kind of inhibit microtubule polymerization and depolymerization. It basically caps the microtubule, if I were to just simplify what's happening here, and it stops it from moving. And the reason that this is kind of important is, as you guys know, every year when you look at Reddit after AP Bio exam is done, everyone's talking about, oh, look, AP Bio expecting me to cure cancer, and that's because a lot of these questions that you see on FRQ do pertain to the phenomena of cancer. And if you know about cancer, it's this uninhibited cell division. And one of the ways that you can deal with that is by inhibiting the cells from dividing. And if microtubules are our spindle fibers, well, we can finally control the toxin level of Taxol to ensure that these rapidly dividing cells within the body cannot polymerize their microtubules, therefore inhibiting the cells from being able to actually divide properly. And that is precisely what we do. And that's because Taxol is what we use to create chemotherapy, right? Chemotherapy, paclitaxel, it's actually a derivative of that Taxol. So I've seen themes that pertain to that idea. So once again, I'm not guaranteeing that you may see that on this year's exam, but it's something worthwhile noting when it comes to cytoskeletal elements. All right, now for the meat, the bulk of Unit 2, which is on cell membrane and transport, again, we'll start here by honing in on the cell membrane a little bit more specifically. We talked about phospholipids, which, by the way, are amphipathic. So they have this hydrophobic and hydrophilic regions, which allows the hydrophobic lipid tails to face each other. And the first thing that I'll say is that, yes, there is this idea of the fluid mosaic model, which you should be familiar with, the mosaicism in that it's not just phospholipids, but you have all of these proteins that are embedded in, and you also have things like cholesterol and glycolipids. And then the fluidity indicating that these things are moving about. They can laterally move very easily. And even in a scientific experiments, we were able to demonstrate that these labeled proteins of the mouse and the human cells were able to integrate relatively well after an hour because of that fluidity. But look, the main thing that I want to talk about is why this phospholipid bilayer can act as almost the perfect substrate through which we create what we call a semi-permeable membrane. Now, in order to understand semi-permeability, I want to start by talking about impermeability, because semi-permeable means that it allows certain things to go through, whereas impermeable means that nothing can get through. And I want to make an argument here that for the most part, when we just take a look at the phospholipids and the things that we care about in biology, this is relatively impermeable, right? In the absence of protein channels and so on. So let's think about why that might be the case from a very logical, deductive perspective, right? So first of all, um, anything that's dissolved on this side is going to be a water. So this side, when I say this side, I mean aqueous solution, a water-based solution here, is going to be hydrophilic. So we're talking about things that are, let's say charged or they're polar. So they like water. And anything that's dissolved on the other side, which is also a water-based solution because of the hydrophilic heads, they're going to be the same thing. They're going to be polar and they're going to be charged substances. But by the virtue of them being hydrophilic, they are going to be chemically inhibited from engaging with the lipid layer here on the inside. So even though this may be just a few thousand atoms thick, I'm not sure exactly what the numbers are, but even though this is a relatively thin membrane, it actually creates this impermeability through the use of chemical signatures, such that anything that's dissolved on one side, in order to get to the other side, would have to go through a lipid layer, which it does not want to do, and vice versa. So it initially establishes that impermeability. And this is incredibly important if you are a cell and you want to ensure that the interior of your cell is distinct from the exterior of the cell. Because remember that the exterior is the environment, and the environment is unpredictable. You don't get to control that. What you can control to the degree that you can is going to be the interior, the cytoplasmic side. And the way that you can do that is by first building this blockade, right? It's sort of like building a wall or having walls around your house or a fence so that you can control what's inside and what's outside. Now, that's the impermeability aspect. But the permeability aspect comes from the fact that we can actually add things like these transport proteins, whether it be these protein channels that you see here. These are facilitated diffusion channels or even pumps. You can actually utilize proteins in their glory because remember, proteins can be shaped into whatever you want because of its flexibility and its structure. You can use these proteins to actually create that permeability for things that may be charged and for things that may be polar. And of course, for larger and larger and larger things, you want to use bulk transport, but that's basically what this entire chapter is going to be about. But whenever I teach this chapter at the beginning of the year, I usually say, look, we're going to be only worried about the transport proteins on the membranes. We're not going to really worry too much about everything that's happening here because this chapter doesn't go into it. But once again, because we're doing this as a review, I will quickly remind you that enzymatic activity, we certainly see that with things like protein complexes of the mitochondrial in a membrane. Signal transduction, we see that in cell communication in chapter 11. Cell-to-cell recognition, we see this in chapter 12 when we talk about how density-dependent inhibition, as in cells that do not divide when there are other cells nearby, well, that's mostly going to be performed through cell-to-cell recognition using this type of structure. Now, it's not exactly glycoproteins necessarily in every case, but at the least, we're going to see these proteins that are found on adjacent cells interacting with each other, typically CAMs. So these are like cell adhesion molecules that are going to, they're going to tell each other, look, I'm sitting right next to you, so you shouldn't divide. And likewise, I'm not going to divide, right? They're able to engage with each other in that sense. So we do see all these proteins come out through the process of studying AP Bio, but for the purpose of this particular unit, is mostly emphasizing the transport proteins, which are involved in the movement of materials. And in order to do that, we learn about diffusion, which again, I'm not going to go into too much because I think you guys are all fully aware of how diffusion works. It's the materials moving from an area of high to low concentration. But we also learn about osmosis, which is actually a lot more emphasized on the AP exam. So, just a quick review of osmosis. Uh, Remember that when you have a system that is connected via a selectively permeable membrane, and you can imagine that this is maybe just a cell membrane here, there are aquaporins, which means that there are already water channel proteins that are always allowing the free passage of water back and forth. So that's sort of why in this image as well, if we're channeling this cell membrane through this membrane here, you can see the movement of water going back and forth. Now, given that the solutes cannot move, what we see playing out in osmosis is that because these are still two connected systems via the movement of water, we have water moving from an area of lower solute concentration to an area of higher solute concentration. Now, oftentimes students ask, do I need to know how to do all the osmotic potential calculations? And in the past, where you had the calculation section on the AP exam, there were every once in a while calculation questions. But in the recent derivation of the exams, I don't think you need to necessarily know how to do the calculations, although, knowing how to do it is always going to be better than not knowing how to do it. But I wouldn't focus on it too much. What I would focus on more is how this pertains to the larger concepts in biology, which deals with things like, well, what happens if an animal or a protist cell is, in an isotonic solution and then move to a hypertonic solution or move to a hypotonic solution. And the way that these questions typically come out is something like this, right? Supposing that an organism lives in a pond that is increasingly becoming saline due to due to the evaporation of water, due to increasing temperatures or something like that. They say, well, what's the problem that they're going to have to deal with? Well, okay, so let's think about this for a moment. So you have this organism that's living in a particular aqueous environment and it's becoming more saline. Now, more saline means that there's greater and greater and greater salt concentration on the outside, right? That's what saline or salinity means. So now what we have is whatever situation that we began with, whichever we're falling under, it's going to be becoming more salty outside. It's becoming more and more and more hypertonic. And as a result, what the organisms are going to have to deal with is the loss of water from the cytoplasm to the outside environment because of osmosis. And that's basically all you have to do. Now, another very big example of this on the exam has to do with paramecia. As you guys know, paramecia are typically thought of as freshwater organisms, as in they live in relatively hypotonic environments. Okay, so let me just kind of reiterate why that is. Freshwater in biology usually refers to hypotonic environments because of the fact that we presume that the cells have a lot of stuff inside. Cells have proteins, nucleic acids, it's got enzymes, it's got sugars. So there's always going to be a lot of stuff on the inside of the cell. If the outside is a freshwater environment, it means that it has less solutes. It's going to be hypotonic. And in the case of paramecia, that's sort of how they evolved, right? This is the environment in which they're actually okay. And the way that they make it okay is by using that contractile vacuole. And as you know, in a hypotonic environment, water moves in. And as that water moves in, it's able to suck up that water through these little tubes that you see there, fill up the contractile vacuole, and they use a lot of energy to actually pump that water out. And they do this constantly. It's almost like the beat of a heart of a mammal, let's say, but instead for them, it's this contractile vacuole that consistently just sort of pumps out that water. And it sucks, right? Like you have to do this your entire life, but that's just the way that it is. Now, one of the things that they could do on an exam is to sort of mess around with this idea that, look, let's say that you take this paramecia that's in its normal hypotonic environment and you move it to an isotonic environment. All right, so you move it to a new solution where you match the amount of solutes on the outside of inside the solution as it is inside the cell. Well, we know from studying this chapter that what you would expect to see is less water flooding in, and that would mean that the contractile vacuole would not have to work as hard, right? But the way that they frame this question is in terms of ATP usage. So, for example, because I told you that contractile vacuole uses a lot of energy, what we should expect to see with ATP usage over time in terms of a graph, let's say. I wonder if I can make a quick chart here. I don't know if, oh boy. All right, so yeah, I mean, I guess I can and just sort of use my mouse. So maybe ATP usage starts off like here, right? Relatively higher. And let's imagine that the x-axis is time that the experiment is performed. And as they move this paramecia into a more isotonic environment, you'll start to see that ATP usage drop. Why? Well, because now there's not as much water flooding into the cell, and you don't have to pump out that water using the contractile vacuole, indicating that the amount of energy that you need to use drops in terms of its background energy usage. And that's something that they love to do, right? Connecting multiple ideas like energy usage and contractile vacuole and the tonicity all in a single question. So I've definitely seen that before. Now, when it comes to the movement of things from an area of higher to lower solid concentration, we call all of that passive transport. And in this case, we see the passive transport of solutes through the channel proteins. In this case, it's a channel protein because it's not a carrier protein that actually changes its shape as it moves things in. But one thing to keep in mind is that whenever we see things like channel proteins and carrier proteins, it looks uncontrolled, as in like, well, is this just going to happen until the solutes equilibrate? Well, not necessarily, because of the fact that channel proteins can actually be closed or opened based on other circumstances that are occurring inside the cell. So in biology, we see things like gated channels. And these are channels that are either gated by ligands. So, for example, they have to bind to a particular ligand to open, or they may even be voltage gated channels, as in they open or close based on the membrane voltage, which again, you don't have to worry about as much for the AP exam without the neurophysio element. But they're not completely uncontrolled, and that's something important to keep in mind. Now, one thing that I kind of glossed over here is that things like carbon dioxide, oxygen, nitrogen gas, so these are non-polar gases, they just go straight through the membrane, so they don't really care, right? So those are what we call simple diffusion of substances, and I've seen questions that come out on that too. So just something to keep in mind. But when it comes to things that are polar or charged, especially things like ions, because we see a lot of ion channels, sodium channels, potassium channels in biology. Well, that's going to deal with a lot of these channels that allow those charged substances to go through the membrane, through the internals of this particular channel protein. Now, we also see pumps. Now, sodium potassium pump is quite important for biology. And I would say that, look, if I were teaching this course in school again, um, I would absolutely talk about this in my lecture every single time. And the reason is that even though sodium potassium channels are typically taught in order to establish a concept called membrane voltage or the resting membrane potential, it is something that can come out a lot on the exam. Just, they explain what it is, they show you the diagram of what it does. It's just better to know it before going into the exam what it is. So essentially, it's a dual pump. I think it's called an antiporter. The idea is that it uses ATP to pump three sodiums out of the cell and it pumps two potassiums into the cell. And the idea is that this creates two separate concentration gradients. So you have the sodium gradient, which is high on the outside, low on the inside, and then you have the potassium gradient, which is low on the outside and high on the inside. So it's sort of reversed. And then the other element of this, which you don't have to know as much, is that because you're pumping out three positively charged ions out and two positively charged ions in, there's going to be a slight negativity on the interior of the cell. So there's a little bit of a voltage potential difference too. But as I said, that's not as important for biology without physiology here. So active transport is important because you could actually use active transport to demonstrate something called the conservation of energy. Now, I know that this is going to be a bit of preview into photosynthesis and cell respiration, but again, that's sort of what we're here to do, right? To connect these earlier units to the larger ideas that we talked about in other latter parts of the course. So let me just demonstrate one quick thing here, and I think that will probably be pretty close to, doing all the review for the first two units. But here we have ATP. Now, hopefully by this time, you know that ATP is a chemical energy source, right? We call this the currency of energy, but it's a chemical energy because the energy is held within the phosphate bonds. And we know that when ATP breaks down and it's going to be an exergonic reaction, that energy can be utilized, minus the heat that's a bit lost. But that energy in this case is utilized to pump these protons from an area of lower concentration to an area of higher concentration. And that's why it's called a proton pump. In this case, pump is really specific to active transport mechanisms. So we have all of these protons that are now on the outside. Now, if I were to ask, well, where's the energy now? Because you know that energy cannot be created nor destroyed. So we started with chemical energy. Where is the energy now? Well, the answer is that now the energy is held within the chemio-osmotic gradient. As in, there is this energy that is held within the fact that these protons are going to want to flood back in. Because if you were to open the gates, they're going to want to flow back in. And there's that potential energy, almost like a rubber band being pulled back, or having water at the top of Niagara Falls dammed and not open yet. There's still this potential energy that once you open the floodgates of the dam, the water's going to come down and you can generate electricity. Or the rubber band, if you let go, it will snap back and it will produce kinetic energy. And in fact, that's exactly what you can do. Because when I use something like a sucrose proton co-transporter, now the chemiosmotic energy that's held within these protons being on one side, as they flow back in, what does that do? Well, that produces energy, or that releases energy that can be used to now drive sucrose from its low concentration to an area of high concentration, which again is a transport work that requires energy. So you can now couple things this way. Now, in the case of, let's say, photosynthesis in the light reaction, or in the ETC of the mitochondria, we didn't use proton pumps with ATP, but we did use things like protein complexes, right, in conjunction with cytochrome C. And what we did was we used either light energy or we used electrons from food to pump those protons outwards. And as they fell back in, they were falling back in through the ATP synthase, and that was creating ATP. So it's sort of a manipulation of this example that we see here. But the main point is energy, as it were, can exist as a function of chemiosmosis, as a function of this set of molecules being stuck on one side, wanting to go to the other side. Another example, and I know that I might be just repeating myself, that you guys might all get this pretty clearly, but it's like blowing a balloon. And when you blow a balloon, all the air inside wants to go out. So you let go of that balloon propels forward. You know that there's energy there that can be released. And that's the main thing that they're trying to establish here. Because right after this, what do you go into? You go into photosynthesis, you go into cell respiration. And there's a reason that the course is set up this way, such that they teach you this in order to establish those foundations. All right. Yeah, and that's basically it, because I'm not going to go through bulk transport. I think you guys can all figure that out. It's not that hard. So you guys want to take a look at some sample questions? Let's do that. All right. So for the last part of today, let's take a look at the last couple of years, and I purposely looked for very specific questions from, look at this, 2024, and this is number 3. So by the way, numbers 1 and 2, those are like the big questions, and 3, 4, 5, and 6 are sort of the medium-sized questions they have now. But clearly, they're still going to be very important. So let's take a look at what they got here. To investigate whether red blood cells of animals lose the ability to take in glucose from their environment as they age, scientists collected RBCs from guinea pigs that range in age from 1 day old to 7 months old. They incubated the equal number of red blood cells in separate culture dishes that contained a 300 nanomolar solution of radioactively labeled glucose. So they're going to try to track glucose as they go into the cells. The amount of radioactively labeled glucose present inside the RBCs of each group was measured over time. So it's a very simple experiment. Again, one of the things that I noticed is that the first part, 3A, you don't really need to, do anything about the text here. This is always going to be just this very broad, general biological fact-based question. Describe a difference between passive transport and active transport. So once again, because we're looking at an FRQ, let me just give you guys a couple of pointers here. Whenever they say describe, use at least 3 to 4, maybe 5 sentences, because they're actually asking you to describe something. So the way that I would describe this, and again, this is being recorded, so I'm sure you guys can, pause and write this down later. But I would say that passive transport and active transport are two mechanisms of transporting materials across the plasma membrane. So I'm setting up the groundwork, right? In passive transport, you have the movement of materials from an area of high to low concentration. Again, that's establishing the main difference, while active transport moves materials from low to high concentration. In passive transport, we see channel proteins and carrier proteins that do not utilize any energy, while active transport typically uses proteins that we call pumps that do use ATP, right? And that's the perfect answer, right? Because what you're doing is you're first laying down the groundwork and saying, look, I know what you're trying to ask me. Like, I know the section that I'm trying to address. And here is the basic background biology, and here is that exact difference that you're looking for. And again, this is an hour and a half section. There are 6 questions. You have enough time, unless you write like extremely slowly. There should be sufficient time because I know that in the hybrid version of this exam, you still have to do FRQ by hand, but you need to at least create each answer as sort of, you know, whole answers, right? You can't assume anything from the reader, all right? Justify why scientists use an equal number of red blood cells in each culture dish as a control. So you guys know, we're just talking about the experimental design and the controls here. So obviously, you want to use the equal number of blood cells because the more red blood cells that you have in one or the other, to what to the same 300 nanomolar solution of glucose. Well, that's not fair, right? Because the more cells you have, the less each cell is going to take up. So that doesn't make any sense. All right. I don't want to be the scientist that has to count the number of erythrocytes, but yeah, well, it's a theoretical experiment. Glucose transporters are required for the facilitated diffusion, again, that's passive transport of glucose into red blood cells. The scientists claim that the expression of genes encoding for these transporters decreases as guinea pigs age. So now we're dealing with unit 6. It's moving on a little bit forward, but that's okay because you guys have all done that. If the scientists' claim is supported by the data, predict the effect of increased age on the amount of radioactively labeled glucose present inside the cells of each group. Okay, so that's a very obvious question. So in the young group, so 1 day old, you should expect to see a lot of radioactivity from the labeled glucose because there should be more of it. But in the 7 months old, you're not going to see as much. And by the way, even for these predictions, at least provide one sentence that justifies why you made that prediction. And you would say, well, you wouldn't see as much radioactivity from glucose in the 7 months old because the older cells would have less gene expression. And you're really just repeating what they're telling you in this question, but it's still worthwhile doing. I think it makes a good statement. Oh, and by the way, sorry. I guess they ask you to justify it anyway in D. So now it's actually even better. Okay, so it's not predict and justify. It's just they have it separately. So that's how you do that. That's pretty easy, right? I'm telling you guys, FRQ is not bad. All right, it's really not bad. You just have to kind of have a good sense of, well, first of all, what it is that they're asking, because chances are, whenever I work with FRQ in the classroom setting, as soon as I explain to students, hey, this is what they're talking about, everyone knows exactly what to say. The problem is that they don't know how to start, right? So it just takes a little bit of practice to see through what they're saying. Okay. Now here's one from, I think this is also from 2024. This might be 2020. Actually, no, this is 2023. This is the cholera toxin one. So now the problem is that there is another image that I didn't include, but I didn't include it because I don't think you need to answer A. Once again, this A says, describe one characteristic of a membrane that requires a channel to be present for chloride ions to passively cross the membrane. Okay, we know that. Chloride ions, by definition, are charged. They're negatively charged. And the membrane, well, what's a characteristic? Well, it's amphipathic, and it's a phospholipid bilayer. And it's got nonpolar lipid regions that are separating the outer and the inner portion of the cell, so the extracellular and the intracellular sides of the cell. So you can't have charged substances pass through the lipid area, so you need a channel. That's it. That's all you're trying to do here. And once again, this might be a couple of sentences, but it's really based on the fundamentals. And by the way, this once again is 2023, right? I'm not going back into 2016 or 2013. These are the last couple of years of the exams, precisely from what we've been talking about for the last hour or so. All right. And explaining why the movement of chloride ions out of the intestinal cells leads to water loss. So now this is about osmosis. Because again, if you have the movement of anything out of the cell onto the outside, what you're telling me is that you're moving solutes from one side to the other, and you are altering that balance of solute concentration on either side. So as the chloride ions leave the intestinal cells, all right, so this is what I was saying. As the chloride ions leave the intestinal cells to the outside, it increases the solute concentration of the outside, which then through osmosis would make it more likely that water through aquaporins will move from the cells to the outside. And by the way, this is talking about cholera. Now, hopefully, none of you guys had to deal with this, but cholera, this is a pretty terrible infection that can cause a ton of dehydration, water loss through diarrhea. And this is basically what they're saying. You have a lot of water loss from the intestines. You're going to get very runny stool. This is what actually kills people. right? And that's what they're saying. And by the way, look at this. We're not even going to look at the other illustration, but we could even do B, independent variable in the experiment. All right. Rate of cyclic AMP production. Oh, I'm sorry. That's crazy. That's dependent variable. Why did I say that? Independent variable. Okay, so that will be whether you have GTP or the cholera toxin in those cells, presumably. Identify negative control. Negative control is anything that has basically the minus minus, or even these can be considered negative controls because you're eliminating one of the factors in a two-factor experiment, and then calculating. Oh, sorry, justify why they use the sample. Okay, so what I'm saying is, um, in many cases, you could actually look at these FRQ and they become so predictable to a degree that even without reading any of the text, like you should be able to answer about 60%. 60% should be answerable, you know? So hopefully you guys have about a month to get there, but yeah, it's not bad. The last question is from a little bit farther back, 2019. In 2019, we have the morning glory petals, and here we're given some pretty descriptive diagrams of what's happening in varying pHs, the flower color, and they tell you that the color of the flower changes as a result of this pigment that's found in the vacuole. And I think what happens is that dependent on the pH, um, right? The color is determined by the pH of the vacuole. You're going to see either the colors being red or blue. And clearly from this illustration, you see that the pH is increasing as the flowers open, and that would mean that the vacuole becomes less acidic, and that makes sense because these protons, which make things acidic, are leaving. Okay. But anyway, so once again, when you see a diagram like this and it seems a little bit overwhelming, don't worry about it. Just go to the question and then just look for the things that you need to look for. And I'll tell you, in 8A, it says, what's the cell component in the model that is responsible for the increase in pH of the vacuole? Okay, cell component that is responsible for the increase in pH. Increase in pH would mean that the decrease in the proton concentration, what is that? That's going to be this one right here. Potassium proton transport protein. That's what's allowing those H plus ions to leave. That's what's increasing our pH. It says describe the component's role in changing the pH. Yeah, so well, it's letting the protons leave, and it's exchanging that clearly with the potassiums here. So it's reducing the proton concentration that increases the pH. That's it. A researcher claims that the activation of this transport protein causes the vacuole to swell with water. Okay, so why is that happening? Well, the potassium going into the vacuole, right, increases the solute concentration of the vacuole, kind of like what we saw with the chloride ions in the previous question, and that allows for osmosis to move this water into the vacuole, and that swells. So that, two things, right? It opens the flower, and it also creates this blue color in the flower. And to be honest, I didn't need to know what this is. I didn't really need to know what that is. I just needed to know this one transport protein and the arrows that are pointing to water going in and a lot of these K plus or potassium ions going in. And that is one of the biggest things that you have to remember. Oftentimes, these FRQ are purposely designed to be very obtuse. They're purposely designed to confuse you. And in certain cases, I've even seen FRQ models where it's just there's just way too many things that it would just take a long time. And maybe it's just my own brain or my limitation, but it's not sensible. There are contradictions inside. It's not easy, but then when you start tackling the question, you start realizing, wait, hold on, I didn't need to deduce everything in that model in order to address these questions. I'm only looking at parts of this model and what makes sense and what connects to the broader concepts in AP Bio. So as I mentioned, I hope you guys can see this a bit of predictability in how these content is being asked on this exam. And of course, as I said, it's going to sort of overlap with a lot of other units, but I try to isolate things for the first two units as much as possible and try to connect what we see here in these two units as they pertain to the larger units in the rest of the course. Okay, so like I said, I think I'm gonna try to do this on live stream next time. I was a little bit unsure about like technical details because I've never sort of had this set up here. I always am downstairs in my studio. Right now, I'm in my office, but it seems to work out okay. So next time, hopefully you guys don't have to stay up till 3 a.m. And of course, if this was a lot more interactive, then I would say, yeah, you guys should, be here in person. But it doesn't seem like it has to be, right? As long as I'm able to deliver what I need to deliver, and you guys don't have too many questions throughout. of course, that's fine. And of course, if you do have questions later on, you can always ask in the Discord chat, right? So you'll always have that opportunity. But that's basically what I had prepared for you guys today. That's right around 2 hour mark. If you guys have questions, um, I'll leave the Zoom chat open for the next few minutes if you guys want to ask anything specifically. But other than that, that's it. I'm sure you guys are pretty tired, especially out in the Middle East. For those of you guys who are out there, it's 4 a.m., I presume. So get some sleep, you know. But that's it. Any questions? All right, that's excellent. Yeah, I hope it's been helpful. And I'll put up another message on our Discord with the next meeting. And like I said, I think it's gonna be like on YouTube, because I just it'll just streamline everything much more better. So, all right, guys, have a great evening. We'll see you guys next time. Bye bye.